All right, welcome everyone. So today we're going to be talking about phylogenomics. So this comes at the end, towards the end of the class. So we have two more lectures on metabolic flux and analysis, and today we're kind of completing the second part of the evolution lecture. So in the first half, we talked about how we can use evolution to study genomes. So we can use evolution to sort of make sense of functional elements in genomes. And we talked about functional elements within proteins, functional elements in non-coding regions, and how we can use evolutionary signatures to distinguish different classes of function between protein coding versus, RNA, microRNA, regulatory motifs, etc. And we also saw how these very powerful deep learning approaches allow us to actually go a step further in traditional analysis of comparative genomics by studying not the changes in the letters, but the changes in the interpretation of the letters. And we focused a lot on the conservation of that interpretation. In other words, even when the letters themselves were changed, we could still see conservation at the level of function, at a higher level of abstraction, if you wish. And that's where all of this representation learning that we've been doing on the machine learning part of the class came in handy. Everybody with me here so far? So today we're switching gears and we're looking at the other part of the arrow. Namely, can we use genomes and many, many species, genes, and alignments to study the process of evolution itself? Okay? And we're going to be talking about three types of analysis at increasing layers of complexity, if you wish. First, we're going to look at phylogenetics, namely, how do we study the changes, not just in an alignment, but in the tree that is responsible for this alignment. So this is going to be taking comparative genomics to the history level, to the series of divergence events. Then we're going to talk about phylogenomics, where you now have thousands of trees evolving inside thousands of trees. In other words, understanding every duplication and loss event beyond just simply can we build an accurate alignment of the sequences that were given. So in the first part of phylogenetics, we're not going to care where the genes come from. But in the second part on phylogenomics, we're actually going to use the species tree along with the gene tree to basically distinguish gene duplication and loss events based on the reconciliation, based on the agreement between these two trees. Everybody with me here? And then in the third part, we're going to, go beyond just the constraint of trees. Basically, in parts 1 and 2, we'll be given a set of regions or genes or elements that we call orthologous or paralogous. And in part 3, we're going to look at how do we even get these to be on top of each other. We're going to look at genome assembly. We're going to look at, all kinds of other ways of alignment. And we're going to also look at evolution very, very near and very, very far. We're going to look at evolution between different hominin lineages or within the modern human lineage, as well as the beginning of life and our innate world. Okay? So a lot of stuff to cover today, but again, this is not on your quiz. The goal is to expose you to all this extraordinary wealth of different types of data sets, different types of questions, many of which are still very active areas of research. Everybody excited? So again, 3 parts. So 3 outlines. And the 3 outlines are going to be number 1, phylogenetics. Number 2, phylogenomics. So we're going to look at reconciliation. We're going to look at gene and species trees. We're going to look at how evolution happens within populations. We're going to look at incomplete lineage sorting and how to blend population and species models. And then we're going to look at the modern synthesis, namely the evolution of evolutionary theory itself. And then in part 3, we're going to look at genome assembly, whole genome alignment, whole genome duplication. We're going to look at human selection, multiple timescales, human demographic history, and also the world before DNA and proteins were even invented. Okay. All right. So that's our goal for today. So part 1, phylogenetics. How do we build trees? How do we score trees? How do we build distances? And so on and so forth. So first, we're going to talk about the basics of phylogeny. Then we're going to go see how we can go from the multiple sequence alignments that we saw in the earlier lectures, in the earliest lectures of the class, to distances by modeling sequence evolution. Then we're going to see how we can use these distances to build trees and tree building algorithms that start from distances. And then we're going to look at tree building algorithms that start from alignments. So we can either build a tree from the distances after having figured out distances from the alignments and modeling evolution, or we can build a tree from the alignments directly, okay? And then we're going to see how, in fact, this is when we build trees from alignments, this is basically just alignment scoring given a tree, but then we have to actually iterate over trees, okay? Let me make this more concrete. But first, we're going to dive in, okay? So what are the basics of phylogeny? So phylogeny is not a new thing. Phylogeny did not start with DNA sequences. Phylogeny started well before, when scientists were looking at fossils and were trying to figure out the history of divergence events that separated these fossils. And those traditional traits were basically morphological data. You would look at a skull of a lizard or a dinosaur or a crocodile, and you would basically say, okay, what are the different parts of that skull? And can I infer phylogenetic histories that allow me to have the least number of events? Is everybody with me here? That allow me to group species together and group fossils together based on the number of trees or based on the shape or based on, whether that particular bone is there or not, and so on and so forth. Okay? So these are what we call traditional traits. These are morphological data. And these traditional traits have very distinct properties from the modern traits of molecular data. So molecular data has a lot of, differences. First of all, we have very large numbers of traits. Every DNA position and every protein residue is a different trait. Okay? These traits are very frequently ill-behaved. Namely, they have back mutations that are very frequent. This is what we call convergent evolution, because most evolution is divergent. You have a common ancestral phenotype and also genotype, and then you diverge from each other. But very rarely would you see convergence, at least in some of those traditional traits. So for example, you can assume that, this particular bone here only evolved once. And therefore, every species that has it is a descendant of that same event where that bone was developed. Is everybody with me here? So this is traditional phylogenies, and our goal in traditional phylogenies was to build species trees. We had a small number of traits. These were hoofs and nails and teeth and horns, and they were very well behaved. Every single event arose once. And therefore, we could use parsimony principle. We could use Occam's razor and basically say, among many different hypotheses that all explain the data equally well, I'm going to choose the simplest hypothesis, namely that something evolved once rather than evolved here, was lost there, re-evolved here, was re-lost there, and so on and so forth. Everybody with me here? So this is Occam's razor, and basically says, if I have multiple explanations, I'm going to choose the simplest one as long as they explain the data equally well. With modern phylogenetics, we have to worry about correcting for back mutations, namely having an A at that position is very easy to re-arise. You just make that mutation again. Okay, so these back mutations where you go from an A to a T and back to an A, there's only 4 options. You mutate, chances are you're going to, sometimes go back. So we're going to have to correct for this. Everybody with me here? Awesome. And then there's also a very small number of letters, and therefore they could arise many times independently. Okay? So now in the lecture for today, we're going to be, or at least the first part, we're going to be thinking about how do we infer a tree from alignments. So alignments, we already saw how we can build, okay? You just basically have this dynamic programming matrix, and you can use that to figure out where should I position the gaps to maximize the score of the alignment, okay? There's, of course, a back and forth between them, because if I have a pairwise alignment between 2 species, then I can use that to infer the position of the most likely ancestor. And then I can use a tree that I can infer to basically walk up and figure out in what order should I be assembling these most common recent ancestors, and so on and so forth. So there's a big interplay. But for now, we're going to separate them and say, we're given the alignment, can we infer a tree? Of course, once you have your tree, you might rebuild your alignment to get a better tree, and so on and so forth. Yeah. No, no. If you have n species, you're starting an alignment. You're starting with an alignment of all n species on top of each other. Here are the positions along a protein, and here's the every row is a species, every column is a letter. So we're starting with that, and then we want to figure out the tree that relates the species. Okay. So basically, the data is going to be sequence data. It's going to be either nucleotide alignments or protein alignments. And then all of our divergence events are going to be binary, even though sometimes we might not really be able to tell whether, say, in a very close phylogeny, whether these two separated first or those two separated first, or, you know, whether this one, etc. But we will assume that everything is binary. And, you know, sometimes we'll have some confidence value in each of these branches that basically tells us, if I resample the positions in my alignment, how often do I get exactly the same topology? Okay. Everybody with me so far? Yes. Awesome. So again, we're going to either first, from the alignment of n rows of different species, first, we can infer an n by n pairwise distance matrix that basically says I already have my alignment, but now I'm going to use this to calculate how far is A from B, A from C, A from D, A from E, B from C, B from D, B from E, etc. And that's my n squared distance matrix. And then I'm going to completely throw away the alignment and only use the distance matrix to infer the tree. Or if I worry about every character, and I don't want to throw away information and summarize it in single distance between every pair of species or sequences, then I will be able to go from the alignment directly into the tree and basically ask how well does that tree capture the events that I observe in my alignment. And then I will couple that with a tree proposal and scoring that allows me to now fine-tune an initial proposal and make it into a better phylogeny. Everybody with me here? Can I get a 5, 4, 3, 2, 1? How well you're following? Okay, super, bunch of fives. All right, so these are going to be the 4 parts of, part 1 of the lecture. So basically, 1, how do we go from sequences to distances? 2, how do we build trees using these distances? 3, how do we go from alignments to phylogenies? And 4, how do we propose trees, topologies? Okay, so dive right in. Everybody with me here? Awesome. So what do we want? We want to figure out how to turn pairwise sequence alignments into pairwise distances. And as I mentioned earlier, there's back mutations, okay? So that's where it becomes interesting. That's where we need a model for sequence evolution. We can't just simply count events, but we need to actually model events. Everybody with me here? Great. So how do we model evolutionary rates? If I see a number of changes, how do I infer a rate from those changes? So let's distinguish different types of events. I could just simply measure how different are the nucleotides. I could use a uniform rate and just simply count the overall percent identity. That's a fine distance. There's been 13% changes. Or I could use transitions and transversions. I could have 2 parameters. One parameter for how often is A changing to G and C changing to T, and one parameter for how often A changing to C or T and G changing to C or T. Everybody with me here? So I could also include within protein coding regions a synonymous rate and a non-synonymous rate. So I don't know if you remember in the last lecture, we talked about measuring not just amino acid changes, but also synonymous changes. And we found some regions that had dual constraint, where you had constraint not just for the protein sequence, but also for the nucleotide sequence. And those were regions where the synonymous rate was in fact very, very low. And of course, we should realize that there are many mutations that don't change the amino acid sequence at all. And those are basically blind, like selection is blind to those mutations. Basically, if the only selection is at the amino acid level, then changing between any of these nucleotides will be fine. So basically changing an AGG into a CGG, both translate to arginine. Okay? And now the last part is that the number of actual mutations is actually typically much larger than the number of observed substitutions. In other words, if I observe 80 events, some of those are likely to be back mutations, okay, where I changed into a T and then back into a C. And then, I have 100 positions. I see that 80 of them are changed. The other 20 might appear unchanged, but some of them will actually be changed twice. And the reason for that is that if I have, for example, a 60% divergence from A into each of the other nucleotides, I have to be actually quite cautious that the next time I change, from an A to a C, the next time I change from a C to another position, I have to be very cautious that I don't change back into an A. Everybody with me here? In other words, for every substitution to be representing only, basically for the only mutations to actually be the observed changes that I see, I have to be very cautious. In other words, most of the time, I will have more events than what I observe, okay? Let me illustrate what that means. Suppose that I have only one parameter. The parameter is how often do I change from any letter to any other letter. So here I'm not dealing with transitions and transversions between, pyrimids and purines, okay? So at every time state, I'm going to start with the letter A, and I'm going to stay at an A with 70% frequency. I'm going to change into each of the other letters with 10% frequency. At the first time point, when I start, let's say time point 0, I'm at 1000. But at the next time point, I'm already at 0.7, 0.11. Also, so far, so good. And then the next one, I'm not at 0.49. I'm not just at 0.7 squared. I'm at 0.7 squared plus 0.01 squared or 0.1 squared, okay? In other words, I can stay at an A by changing to a C and back into an A. And therefore, my probability of changing, of staying in A is not just 0.7 squared, which would be 0.49, but instead it's 0.49 plus 0.1 times 0.1, 0.1 times 0.1, 0.1 times 0.1, where I basically switch from an A to a C and back into an A. That's 0.1 squared, that's 0.01. At short evolutionary distances, it doesn't matter at all, or at least it matters very little. But as I go further and further and further, say I get to 10%, 20%, 30%, 40% observed changes, then if I observe 40%, I've actually done almost 60%, okay? So if I observe 10%, I've actually done 11. If I observe 20, I've actually done 23. If I've done 60% change, I've actually changed nearly every position on average, okay? And if I observe 70% divergence, I have on average 2 substitutions per site. Everybody with me here? What if I observe 80% divergence? Who can comment on 80% divergence? How far do you think that gets? Now, what does 80% divergence mean? How many letters do I have in the DNA? 4 letters, A, C, G, T. If I take a random sequence and I put it on top of another random sequence, what % difference I'm going to observe? Good. The limit is basically 75. If I have 80% divergence, I'm basically doing something wrong with my alignment. Okay? My alignment basically sucks. If I have a good alignment algorithm, there's no way I'm going to be able to align 75% or more than 75%. And the reason is that the aligner itself is going to just like move some locations and it's going to say, well, I have 3 A's here, I have 3 A's here, I'm going to move them together. Is everybody with me here? So basically, I saturate at 0.75, and I basically hit infinity. Okay? So basically, at that point, you hopefully can see that, like, as I go further and further and further, I asymptote towards 75% divergence. Sounds good? And that basically means that I need a way to correct. I need to basically say, okay, I never have the actual, I only observe mutations. And therefore, I need a way to correct from the observation to the actual number of mutations. And that involves simply taking that equation, expanding it out, and just saying, as I move further, my forward process is going to be that the number of, conserved amino acids is going to be 1/4 times 1 plus 3 e to the minus 4 at, which is simply if I run the stationary process to completion, or at least to time t, then I'm going to basically end up, if I run this Markov process, I'm going to end up with, some curve. And then I can just read off the curve what the actual number of mutations is by reversing this equation. So this is, the number of conserved and the number of substitutions. And now the reverse process simply tells me, here's, how do I solve for t, given that I have this? And then the actual distance is going to be, 3/4 log of that, which is reversing that equation. Is everybody with me here? So I have this asymptote, this exponential driving towards 0.25. And then I need to reverse that exponential to basically figure out what's the actual. And therefore, if I observe, 0.3, 0.4, 0.5, etc., I can infer this. Can I get a 5, 4, 3, 2, 1 who's following me here? Awesome. Great. Any questions? Can I get everybody's hand up, please? Okay, awesome. Good. All right. So that's the simplest model. This is known as the Jukes-Cantor model, and it has only one parameter. And that's basically the part about actually modeling evolution. So how do we turn pairwise sequence alignments into pairwise distances? What we need is a probabilistic model of divergence. And the simplest one is the Jukes-Cantor model. Everybody so far? Awesome. So that's the simplest model. I can be a little more complicated. I can basically say that A to G and C to T change with each other at a transition rate, which is, at the transversion rate, which is less frequent than the transitions between, the colors are off here. So A to G are transitions, C to T are transitions, A to C, A to T, etc. The rest is transversions. Okay? So basically, the substitution rate between purines and between pyrimidines, or within purines and within pyrimidines, is actually much higher than the substitution frequency between the two categories. And therefore, I will have two parameters, and I will need to estimate those two parameters based on the observed mutations. Okay? And then this is the simplest model, where I have only one substitution type. This is the Kimura two-parameter model, where I have one transition parameter, one transversion parameter. And all of these are assuming equal base frequencies. I could then have two different transversion frequencies, one between, say, A and C, and one between, A and T, or something like that. Or I could have a fully symmetric model, basically where I have, 1, 2, 3, 4, 5, 6 parameters, or 9 parameters, or something like that. And then I could basically have all of this is with equal frequencies, and I can also have with unequal base frequencies additional substitution types. Is everybody with me so far? So this is all about modeling sequence evolution, okay? So now I've basically aligned my sequences. I've figured out all of the distances between every pair by simply counting all the alignment. I remove any kind of joint gaps, which are gaps on both. And I simply count for every position that's on top of each other, what is the actual distance based on the aligned positions. For now, none of these are dealing with gaps. None of this is dealing with sort of fancier events like duplications, losses, or segmental changes, etc. It's just counting the number of events. Okay, question? Okay. Everybody with me? Okay, now let's look at how we can go from distances that I've just computed using my model of sequence evolution to actually trees, okay? So I now have my n squared matrix of distances, and I want to build a phylogeny. What does that mean? That means that I want to figure out a tree such that moving from species A to species, I don't know, M over here, or species P over there, along the tree is recapitulating the distance that I have between species A and species M, or species P. Is everybody with me here? So to be able to infer a tree that matches, quote unquote, my matrix of distances simply means that I will be able to compute any pairwise distance by basically just walking up and down the tree. Try to figure out the distance between human and cat. It's this branch plus this branch plus that branch plus that branch. And if that matches exactly the tree distance that I had between the matrix distance that I had between human and cat, then I'm doing great. Can I get a 5, 4, 3, 2, 1 on this concept? Awesome. So that's basically fitting a tree to a matrix. Sometimes it's possible. Sometimes it's not possible. And when it's not possible, we're going to have to figure out what is the best tree that matches those distances. Okay? So we have the goal of minimizing the discrepancy between the observed pairwise distances, which are completely unconstrained. I don't have to walk along any roads. I can just go from the dog, I don't know, house to the rat house by teleportation, which costs me the dog-rat distance. Or I can walk along the paths that connect all these houses, and then I have to pay the tree distance. Okay? Everybody with me here? Great. So now, when the matrix is such that it can be perfectly encapsulated in this sum of paths, then that's known as an ultrametric distance. And moreover, actually, I should say that that's known as an additive distance. So an additive distance basically means that I can just add up all of these distances, and then my matrix was basically generated by adding up these segments. Everybody with me here? Now I can impose an additional constraint, which is that all species evolved at the same rate since their common ancestor. So basically, I can fold up the tree in a way that says that the trees actually represent divergence times, not just number of events. And therefore that, I don't know, 65 million years ago, you had a divergence of, or like 90 million years ago, you had a divergence of all the mammals. Then, I don't know, 65 million years ago, you had a divergence between human, mouse, and rat. And then, I don't know, 30 million years ago, you had a divergence between dog and cat. Is everybody with me here? So if the matrix is such that it can be represented as a series of times elapsed since a common time of divergence, then that's known as an ultrametric distance. Okay? So an additive distance is simply a distance that can be built by a tree, and an ultrametric distance is simply a distance that can be made by traversing from a common ancestor down to every branch with the same exact number of steps. So far, so good? Good. So again, an additive distance can be checked simply by satisfying this 4-point condition. But all the 4-point condition says is that for any 4 points, I can find a midpoint branch that, satisfies this. And notice A and B don't need to be the same. Whereas for an ultrametric tree, every single time I have 3 points, there is, 2 of the distances are always identical. And the reason is that this must be a tree. And guess where's the root? Exactly at the midpoint. Okay? But of course, most of the time, I'm just going to have general distances. Okay? So if I have an ultrametric tree, in other words, if the distances were indeed generated by a clock model, where every single one of these species diverged at a specific time from the common ancestor and from today, then I can basically use UPGMA, which is unweighted pairs grouping with arithmetic mean. So UPGMA is effectively a clustering algorithm that says I'm going to find the two closest species, group them, and put the midpoint. I'm going to find the next closest, group them, and put the midpoint. I'm going to find the next closest, group them, and put the midpoint. I'm going to find the next closest, group them, and put the midpoint. And if indeed my tree was generated by such a process, then this algorithm is guaranteed to find the correct tree. If it wasn't, absolutely no guarantees. So far, so good? Okay. So this is UPGMA, also known as hierarchical clustering, basically unweighted pair group method with arithmetic mean. And it's what I just said. You just take every closest pair, group it, make a cluster out of it, etc. And then in the case of, human, mouse, rat, dog, you basically just keep, averaging, and then you build a tree progressively, and then you're very happy. Okay. Now, there's a problem with UPGMA. It assumes a molecular clock. And it implies that the time is constant for all species. And therefore, in a case where the correct tree has some species that accelerated, for example, I don't know, this is a tiny little species, it migrated to a new climate, it now needs to adapt very rapidly. Or this is a very slow generational species, every species lived till 300 years, and then, there's very few mutations. Okay, let's say that's a turtle, and that's a, I don't know, chihuahua or something. Okay, everybody with me here? So if the correct tree looks like this, what's UPGMA going to do? It's going to assume that they were generated by a clock. And therefore it's going to say, oh, well, 2 and 3 are clearly the first ones I should merge because they're the closest. And their midpoint is exactly in between them. And then the next one is going to be, I don't know, 4. And I'm going to group that there, etc. So UPGMA is basically guaranteed to have the wrong tree if there are some branches that are shorter and some branches that are long. So far, so good? So that's where neighbor joining comes in. Neighbor joining says, okay, well, why don't I deal with this situation? How am I going to deal with it? By saying that if a species is repeatedly far from all of the other species, I'm just going to assume that that species is antisocial and has a long branch. And if a species is repeatedly close to all the other species, I'm going to assume that it's very promiscuous and it has a very short branch. So I'm going to do a correction factor that basically says, what is the sum of distances from my species to all of the other species? Let me just simply correct by that when computing my distances. So I'm going to have a promiscuity index for every species. And I'm going to adjust my distances to be revised distances, where I'm just going to subtract the promiscuities of the two distances from that distance. Is everybody with me here? Can I get a 5, 4, 3, 2, 1? Awesome. Good. And that's what neighbor joining is all about. And the idea here is we're going to define the set of leaf nodes. We're just going to sort of progressively merge, but using this refined, distance. Okay. And that's great for the additive distances. In other words, neighbor joining is going to do fantastic for additive distances. UPGMA is going to do fantastic for ultrametric distances. But most of the time, we're going to be here in the general distance scenario. Okay? So what are we going to do in the general distances? Well, we can still use neighbor joining. And that will probably be a fine tree most of the time. Or we could be a little fancier and say, well, let's define an optimality criterion. And that optimality criterion could be either character or distance-based. I could basically say, maybe I want a least squares solution, where I can basically simply see what is the deviation that I have from adding these distances, between adding these distances and then the original matrix distances. And I'm just going to simply set all of these parameters to minimize the sum of square distances between what I get for each of those. Is everybody with me here? So then I have an optimality criterion, which is basically I want to just minimize the distances. And then I'm just going to have some kind of fitting algorithm that's going to try to select these. And, maybe I will have some kind of gradient descent that basically says if I adjust this distance, how is it changing? And so on and so forth. Is everybody with me here? Awesome. So that's in the general case where basically, we could minimize the distance discrepancy with least squares or with minimum evolution. Okay. As for character-based, well, let's talk about it next. Okay. So what have we talked about? We've talked about the basics of phylogeny. We've talked about how to go from alignments to distances, how to go from distances to trees. We looked at different tree types: ultrametric, where there's a clock; additive, where it was generated by a tree; and general distances, where all bets are off. We talked about UPGMA, that works great for ultrametric. We talked about new neighbor joining, that works great for additive. And these have guarantees, but also have limitations in the case of the general distance. And we also talked about this optimality idea of maybe we could adjust the tree branches to minimize the least squared error or minimize evolution. Okay. And then again, all of these require a search, which is what we're going to talk about number 4. Okay. So now let's talk about how we can go from alignments directly to trees. So how do I go from an alignment to a tree? Okay. Everybody with me here? So what are we going to do? We're going to look at different approaches for doing that. The first approach is parsimony, where I could be either set-based or use dynamic programming. And then the third approach is maximum likelihood. Okay. So again, we saw how we can go from sequences to distances, from distances to trees, or from sequences directly to trees. And that's what we're going to do now. But remember that everything we're going to say now in section 3 is for a given tree, how well does it match a given alignment? But then we're going to sort of have some tree proposal and scoring coupled to that. Okay. So character-based phylogeny is essentially about scoring, not necessarily about tree finding. So we need to couple them with a tree finding algorithm. Okay. So, and that tree finding algorithm is going to be some kind of tree proposal and update, and that's going to be part 4. And there's 2 approaches which use the same architecture. One seeks to minimize events, and that's parsimony. And the other one is probabilistic, and that's maximum likelihood or maximum a posteriori. We have an alignment. An alignment is made out of many columns of the alignment. So suppose that we're looking now at one of the columns of the alignment. Everybody with me? So in one of the columns, I see B for one species, A for another species, B for one species, A for another species. And I want to know how bad or how good is that tree for capturing the states of these variables for each of the species. Okay? So how good is it? Well, if this is my tree, I have to infer a cost. What is a cost? A cost is the number of events. So I can say, here's how many events I need to explain my tree. And to get that, there's a very simple algorithm, which is a parsimony algorithm. That basically says I'm going to look at the nearest neighbors, and I'm going to walk up the tree, and I'm going to infer the most likely, or at least a good ancestral state for every one of those. So if I have an A and a B here, what are the possibilities for the ancestor? Either A or B. Okay, so I'll just choose the set A or B. And what is the cost? The cost is 1, either because I changed that ancestral A into a B here, or because I changed that ancestral B into an A by mutation event along this branch. 5, 4, 3, 2, 1. Who's following with me? Awesome, great. Now, if I have an A or a B at that position here, and I have an A here, what should I put there? Probably A. Okay? So then the next step after that is if I have a non-empty intersection between the set of possible characters here and the set of possible characters here, then I'll just take that intersection. Okay? And then if I have an A or a B here, then I'll take the union again. Is everybody with me here? So this is the simplest possible algorithm. If the intersection is empty, then I choose the union. If the intersection is not empty, then I choose the intersection. End of story. Yeah. So this could be an A, a G, an A, and a G. So here I've conflated the concept of a species and a character. In this particular case, I'm looking at one column of the alignment. And in that column of the alignment, I have character A, that could be rounded hoofs. Character B could be pointy hoofs. So I have rounded hoofs, pointy hoofs, rounded hoofs, pointy hoofs. Or it could be the actual letter A, G, A, G, and so on and so forth. These are all great questions. So, don't feel bad for asking something that probably other people were wondering as well. Okay. And then as for the species, yeah, you could call this bucket the human bucket, the chimp bucket, the rat bucket, and the mouse bucket or something. Okay. Everybody with me here? Awesome. So there's a problem. The problem with this union intersection approach is that it doesn't actually capture all possible trees. In other words, it will only explain this particular tree by having an A at that position. But when you look further up, you basically realize that, wait a minute, there's a B here and there's a B here. So I could have gone B all the way and then just change into an A once and an A twice in this particular scenario, okay? This is equally optimal. It's still only 2 events where the ancestor was a B, but it's simply not accessible to the union intersection approach because it doesn't look ahead, okay? So what are we gonna do instead? We're gonna use dynamic programming. What dynamic programming allows you to do is to remember all of the possible scores for all of the possible paths, find the maximum score, and then trace back to find the optimal path. In other words, it will postpone making that decision until the end of the search. So instead of just saying, oh, there's a non-zero intersection, great, I'm going to just set it to that. It's going to maintain all 4 characters. So it's going to say, at every position, I can be an A, C, G, or T. And my observable species are mouse, rat, human, and dog. So my mouse costs, if I set the mouse to A, my cost is 0. If I set the mouse branch to C, G, or T, my cost is 1 because I need to change it from that back to the observable A. Is everybody with me here? So I can do that for the observed variables, mouse, rat, human, dog. And I can just set it to exactly what's observable. The one that's observed is the 0 cost one. And now, at every merge, I just simply add up the costs. So at position B1, if I set B1 to be an A, then I have a cost of 1. If I set B1 to be a G, I have a cost of 1. So in both cases, I have an optimal cost, and I'm maintaining the fact that there's 2 possible solutions, both with cost 1. One step above, I now have the costs for B1, and I just simply add up the cost for the human. And then the cost for B2 is 1 if it's all set to 1, or 3 events if I set it to C, 3 events if I set it to T, or 2 events if I set it to G. So far, G doesn't look good at all. But at the next iteration, when I go to B3, based on the states and costs of B2 and the state of D and cost of D, then at B3, 1 now becomes a 2, but then the 2 becomes a 2 again because there's no cost. And suddenly I have 2 optimal solutions. One is an A and the other one is a G at that ancestral node. Can I get a 5, 4, 3, 2, 1? So who's following? Awesome. Great. So that is a parsimony approach, but using dynamic programming going up the tree. Okay? And that's actually going to be the foundation for many of the other approaches. This concept of dynamic programming to basically infer a cost function for every possible state of the intermediate variables. Okay, great. So we basically looked at a greedy approach of union intersection that doesn't access all solutions, but still is guaranteed to find an optimal score, or a dynamic programming approach that actually sums up the costs as it goes up the tree. Okay, now let's look at a maximum likelihood and a maximum a posteriori, more probabilistic approach that also includes back mutations, etc. Okay, so what are we going to do? We're going to build a generative model. And that generative model is going to have some kind of process through which it is generating trees. I can have a duplication loss process that generates the topology, and I have a mutational process that generates the actual sequences given the topology. That basically says that I will have some maximum likelihood interpretation of the sequence alignment data D given the branch lengths B and the topology T with which I am generating my sequences. So I'm sampling a set of data, I'm sampling characters using a topology of a tree and using branch lengths. And what I want to do is maximize, basically do a search over all possible branch lengths and topologies to find B hat and T hat, the optimal branch length and the optimal topology that best explain my data. Can I get a 5, 4, 3, 2, 1 here? Awesome, beautiful. Alternatively, I could say that I don't want to just maximize the probability of generating the data given my branch and topology. What I want instead is to scale that by a prior, this should say prior. I want to scale that by a prior of what are some completely unrealistic branch lengths and topologies. Okay, so I want to have a Bayesian approach that basically says, what is my prior over all branches and topologies, and what is the likelihood? And together, I'm going to get the posterior. Okay, another way to think about that is the posterior is having observed the data, in other words, posterior to observing the data, what is the maximum probability, branches, and topology? Okay, and this is just using Bayes' rule. Can I get a 5, 4, 3, 2, 1 here? Awesome, beautiful. Okay, so that's the likelihood of the data given the model. And then this is the prior on the trees and branch lengths. And how do I generate this? I can have some kind of duplication loss model. I can have a birth-death process or a Yule process and so on and so forth that generates topologies and branches. And here, if I know the branches and the topologies, then the probability of generating my data, i.e. the sequences, is simply the evolution model that we looked at before. Namely, I have a forward process that generates data according to some probability. And I can just say, given a Jukes-Cantor model, a Kimura two-parameter model, HKY model, what is the probability of generating the observed sequences? Why is that helpful? Because I can say, given topology A and topology B, which topology better explains my data? Okay, and that probability is going to be what I'm going to be using to choose the maximum likelihood or the maximum a posteriori tree. Everybody with me here? Who feels that they're learning stuff? Yeah? Good. Again, this is taking a lot of concepts that we've seen already and just applying them in the context of phylogeny. So we can compute that recursively using the exact same idea of dynamic programming that we did before. In other words, instead of just doing a very simple score of +1, we're going to do a slightly fancier score as I go up my tree. Everybody with me here? All right. So what are we going to assume? We're going to assume that every column of the alignment evolves independently. It's a very simple assumption. We're going to treat that column in isolation. And then we're going to do the product over all of the columns. Okay? Everybody with me here so far? That's step 1. Step 2, we're going to assume that every branch evolves independently from all other branches. In other words, it doesn't matter what happens in T6. T7 is going to have its own evolutionary process. That's fine as well. So we're going to condition on the parent and just say, if I've observed the parent, I don't care what happens in T6. I'm just going to evolve down my T7 branch. Okay? So I'm going to expand the total probability into some product of each of the children conditioned upon its parent and the time elapsed along that branch. So far, so good. And the only thing that remains is this probability of an ancestor, which is some kind of root prior, which is basically just a background nucleotide frequency. Now, how do I compute each of these branches for a single column? It basically says I have a G here and I had an A here. That's just my Jukes Cantor or cumulative parameter model and so on and so forth. Just a single event, or at least a single branch, isolated from all other branches. I know the parent, I know the child, and I know, how many changes there were. So how do I compute the probability of a child given the parent and the time? Well, that's just, the model that we have already, and we can easily calculate it for any given assignment of the internal node. And because the internal node values are not known, they're unknown, we're going to basically marginalize. What we're going to basically do is we're going to compute the dynamic programming algorithm going up that tree, up these branches, maintaining the uncertainty, and we're going to sum over all possible values of all internal/root nodes, and that will be what will represent the sequences of all of the internal nodes. Okay? So basically, the joint probability of observing all of my sequences, X1 through X5, aligned to each other, given my topology and the branch lengths, is going to be simply that in isolation, which we're given this branch independence, we're going to assume that it's just going to be, each child given its parent and the corresponding branch length, which is going to be simply that product. Can I get a 5, 4, 3, 2, 1 here? Awesome. Beautiful. All right. So again, side evolution of a single sequence. We know how to do that. Evolution of a single branch. We know how to do that. Just multiply all of the sides together. Evolution of the entire tree. We just walk up the tree. And then marginalizing of the hidden ancestral sequences. This is basically the dynamic programming matrix, which I set up exactly like before. And when I put it all together, I have this very efficient marginalization over all of the tree. And I have this dynamic programming algorithm for, computing the scores for every one of the characters in every one of the ancestral nodes, which I'm going to be adding up according to this case analysis. And in the end, what do I end up with? I end up with scores at every position. And what did I do for my very simple sum before? I just took the maximum score and I just ran down with it. Okay? Just like before, I basically had my minimum score, my minimum cost to here. I set that value to A, and then I basically said, okay, well, how did I get here? I need to remember the arrows. I don't know if you guys remember that from dynamic programming earlier in the course, but basically every time I say I got a 2 here, I'm going to remember what were the states that gave me that 2. And I'm going to do the same thing here as I go back down the tree to basically compute the overall cost. Okay? So we're going to fill in all of the internal node likelihood vectors. When we reach the root, we're going to multiply the base frequencies, and that's going to give me all of the states, and I can kind of go back and compute that. Okay, so this is this third part here, where I basically said I can go from sequences to distances, I can go from distances to trees, and I can go from characters to trees, score different trees, and figure out which tree gives me the best likelihood or the best a posteriori probability. But now I need to actually suggest some trees. How many trees are there? It's an exponential number again. Because for every one of them, I could sort of choose which one of those I want to go up. And then that gives me, way too many choices. So what are we going to do instead? We're going to basically iteratively refine the trees using some kind of stochastic process for walking through tree topologies. So I'm going to initialize maybe with a neighbor joining tree because that's a fine algorithm. Works well a lot of the time. And then I will compute the probability of that tree, given what we just talked about. And then I'm going to modify the tree using some kind of operations. What are these operations? Well, I could do nearest neighbor interchange. That basically says, I have A, B, C, D this way. I'm going to change it so that it becomes B, D, A, C. Sounds good? So that's a nearest neighbor interchange event. Or I can do subtree pruning and regrafting. That basically says, well, C is connected here. I'm not going to move it to connect it here, and so on and so forth. Okay? So, and then I'm going to score the likelihood of the modified tree, compare it to the likelihood of the tree that I had before. And if it's better, I'm going to keep it with some probability. If I keep it all the time, if it's better, then it's basically some kind of greedy hill climbing approach, and I'm going to end up at some local maximum. If instead I keep it with some probability, that means that I might be able to get out of this local maximum, okay, and explore more of the space. Moreover, if I keep it with a probability that's exactly the difference in the likelihood ratio between the two trees, then I guarantee that I will be exploring my space according to the posterior probability distribution of all tree topologies. Okay? And that's what we call Markov chain Monte Carlo. It basically says I can take any proposal rule as long as it's connected, that as long as I can explore every topology from nearest neighbor interchange and subtree pruning and regrafting, I can take that proposal rule, and when it's worse, I could basically decide to take it or not take it. When it's better, I can decide to take it or not take it. And based on exactly the way that I do these decisions, that I make these decisions based on the probabilities, I can basically estimate that posterior probability distribution, effectively sampling from the universe of tree spaces in a probabilistic way. Okay? The last component is that I want to estimate some kind of reliability, because every single time I have an alignment, that alignment is a sample. Every time I have a species, that species is actually a sample. Every time I pick an individual from that species, that individual is a sample, and so on and so forth. So what I want to know is if I had chosen a slightly different alignment, if I had chosen a slightly different part of the genome, if I had sequenced a different gene, would the topology be different? So for that, what I can basically do is resample columns of my alignment and redo this type of operation at those different columns. Okay? Awesome. So that completes part one of this very ambitious lecture. That basically says, how do we build phylogenies? We basically can go from sequences to distances, from distances to trees, from alignments to trees, as long as I have these tree proposal and score. Can I get a 5, 4, 3, 2, 1 on this part? Awesome. You guys are great. All right. So this was part one. Let's go to part two. Part two basically says that up until now, we've only worried about one type of object. It didn't matter if we were comparing species or genes or, every hemoglobin or only one ribosomal sequence from each of 100 species, etc. Or, God forbid, a bunch of hemoglobins from a bunch of mammals. So suddenly, I can do a lot more if I have knowledge of what every element in my tree represents. Okay? And that's what we're going to do now. Now we're going to deal with phylogenomics. The first part is to basically realize that gene trees evolve inside species trees. What does that mean? That means that every time that I have a speciation event, say between mouse and rat, the mouse is walking around with a bag of 20,000 genes. As soon as it speciates from rat, all of the genes that it carries have to speciate. Everybody with me here? So basically, that every gene tree, if I label the branches, remember before, I hadn't even labeled my branches, but now I've labeled my branches, dog, human, mouse, rat. And if I build a tree topology that is discrepant from the species topology, then I have to imply that there were some duplication and loss events. Okay? So that basically means that if I have any kind of gene tree that I'm trying to study, I can label the corresponding branches according to the species that they came from, whether that gene was sequenced in a dog, in a human, in a mouse, in a rat. Okay? And now, if I have a duplication event, as I mentioned earlier, all of the 20,000 genes in the bag of mouse have to diverge. Sorry, if I have a speciation event, all of them have to diverge. But now if I have a duplication event, that duplication event means that on the common ancestor of mouse and rat, that gene was duplicated. What does that mean? That means that at every speciation event, the two copies are going to be speciating. Okay? So that basically means that all of the descendants of that duplication event will now have two copies. Is everybody with me here? Awesome. So let's make some definitions. A gene in dog that has one and only one copy in human that fulfills the same function and that arose from the same common ancestral gene that also fulfilled that same function is an ortholog. Ortho means right up, straight up in Greek. So ortholog basically means it's a gene that descended by vertical descent from that other common ancestral gene. Now, if I have a duplication event and I now have two copies, say, hemoglobin A and hemoglobin B, those are paralogs of each other because they arose from a duplication event. And paralogs have all kinds of properties. Paralogs might have different functions. Paralogs might take on new functions. Paralogs might each keep a subset of the ancestral functions of the pre-duplicated gene. Is everybody with me here? And paralog means on the side. So ortho means straight down, para means on the side. Everybody with me here? So an ortholog and a paralog can basically be defined functionally, just like I did. Ortholog has the same function. Paralog, arose from gene duplication. And I can also define them based on their phylogenetic relationship. Now pay attention here. An ortholog pair is a pair whose common ancestor is a speciation event. A paralog is a pair whose common ancestor is a duplication event. What does that mean for R1 and R2? Trace the history of R1 and R2. R1 and R2 coalesce. Coalescing basically means as I walk up the tree, I meet. So they coalesce at a duplication event. Everybody with me here? So that means that they're paralogs. Now, what about M1 and R1? Are they orthologs or paralogs? Shout it out. M1 and R1. They coalesce at a speciation event. Therefore, they're orthologs. M2 and R2 are orthologs. But let me give a tricky one. M1 and R2, what are they? They're in different species, but they're different copies of these genes. In fact, R2 and M1 coalesce at a duplication event. And therefore, M1 is a paralog of both M2 and R2. R1 is a paralog of both R2 and M2. But R1 is an ortholog of M1 and M2 is an ortholog of R2. Everybody with me here? Awesome. So now I want to define what are orthologs and what are paralogs. For this, I better have A, a correct phylogeny, and we're going to worry about that. B, a correct species tree, and we're going to worry about that as well. And C, a correct mapping of my gene tree to my species tree. And let's talk about that now. Okay? So how do I map a gene tree to a species tree? This is known as reconciliation. Basically says that I have a bunch of branches labeled by species, and I'm going to reconcile them on the species branches. That's not obvious. The way that I'm going to do that is I will basically map every single one of them as low as I can go, but no lower. Okay? Basically that simple. M and R, I'll map it to the mouse rat common ancestor, but no lower. MR, same thing. This one, same thing. This one, well, what do I have underneath? I have DMRMR. So basically, I better map it all the way there, but no lower. Everybody with me here? So that's basically a very simple explanation for what reconciliation is. So I basically build my species tree, I build my gene tree, and then I reconcile them by simply mapping to the least possible height. So I recursively solve this particular algorithm, which at the root of it has this least common ancestor. In other words, the most recent common ancestor of all of the branches, of all of the species that are represented in the children of that node. Can I get a 5, 4, 3, 2, 1 here? Yes, awesome, beautiful. Okay, so that's the reconciliation problem. And now here's where it gets tricky. If I reconcile the wrong way, if, for example, M and R reconciled here by mistake, then that implies all kinds of duplications and losses. That's why a reconciliation algorithm is very important. Okay? All right. Now that we have distinguished gene trees from species trees, we can do all kinds of new cool things. Okay? So first of all, if I'm trying to build a correct species tree, and I'm going to color those blue as always, then I could average a bunch of gene trees together. In other words, I'm trying to figure out in what order did a bunch of mammals diverge from each other. Is the red panda closer to panda, or is it closer to a bear or a raccoon, and so on and so forth? Okay, so how do I do this? Well, I can take every gene in all of these mammalian species and build a topology for each of those using the methods that we saw in the first part of the lecture, part 1. Then I can somehow combine them to infer the species tree. How can I do that? Well, there's again many methods. I could use a super matrix method that basically looks at the matrix of relationships and then, figures that out. Or a super tree method that looks at the set of all possible splits of two branches. I could minimize the deep coalescence. That basically means that as I walk up my tree, that all of the pairs coalesce as close as possible to their branch, but they do not traverse species divergence events. Or I can explicitly model coalescence. And I can use that to basically infer the most likely scenario for all of these ancestral trees. And again, I want you to sort of get out a key insight here, which is that I can represent every split between a group of n species in this n squared matrix that basically tells me what are all of the possible splits. And then I can count every topology as simply what split does it induce if I look at that topology. Okay, so the way to reconcile topologies is by start thinking that every branch simply separates AB from DC. Every branch here, AB from CDE, and so on and so forth. So even though every gene tree could have a different subset of species represented, the splits that it induces can be compared across these different topologies. Everybody with me here? Awesome. All right, now we should basically realize that I can actually use this information to build better gene trees. I can use this information to now have a better model for how gene evolution happens. Remember before I was sampling topologies, I was sampling branch lengths. I can now have a much more sophisticated model if I understand that there's species trees and gene trees. What are we going to do now? We're going to basically build a generative model for learning what a gene tree should look like if I already know the species tree. So we're going to use the concept of a common species tree across thousands of gene trees to improve gene tree reconstruction. Everybody with me here? So what we're trying to do? Trying to leverage this common information to learn some common properties of the evolution of genes in that lineage of species and use that to improve reconstruction. In other words, if I have a known species tree of dog, human, mouse, and rat, and I end up with a topology of my gene tree where the human diverges before the dog, okay? So that lineage here diverges there. I've just swapped two branches. Doesn't sound like a big deal. But swapping those two branches infers that there was, in fact, an ancestral duplication event in the common ancestor of those four species, followed by speciation, another speciation, and then a loss here, and so on and so forth. Okay? So basically what I need is 1, 2, 3 losses and 1 duplication, which are very costly. Whereas the cost difference between, branching here or branching right above that might be very small. Is everybody with me here about the intuition? So therefore, if I know something about the species topology, that can constrain my gene topology. Do I always want to follow the gene topology? No. But when there's sufficient evidence, I'll be able to overrule the species topology. And that's what we want to model. Everybody with me here? So this is what we call phylogenomics. And this is something that my student, Matt Rasmussen, and I developed, a few lifetimes ago. So we want to develop a rates model that basically said, what is the probability of a given tree given, number 1, the topology, number 2, the reconciliation, and number 3, some parameters that are representing the evolutionary rate of that gene, okay? So what did we notice? We noticed that as we took different genes in the Drosophila phylogeny of 12 species that we had, different genes basically showed a scaling that was uniform across all of the species. Why? Because some genes are very constrained and other genes are very fast evolving. But when a gene is constrained, it tends to be constrained similarly across all of the different species where it evolves. And when a gene is fast evolving, it tends to fast evolve in all of the lineages where it's evolving. So everybody with me here with this concept? So that's the gene-centric view, which basically says that this gene will evolve fast or slow. Let me now take a different view, which is the species-centric view. This species here has a very long branch because it evolves rapidly. I don't know, it sort of was separated by a volcano. It suddenly adapted to a new environment. And all of its genes were more rapidly evolving, or that species has a shorter generation time or a slower mutation rate. So you could think of it conversely as there's an overall species tree, which is a normalized version of all of the gene trees, where this species here is always longer than those 3 species here. And it doesn't matter what gene I throw in there, that gene will end up evolving slower in that lineage. Everybody with me here? So again, these are the 2 forces. The 2 forces are, on one hand, you basically have a species rate, and on the other hand, you have a gene rate. Okay? Let me put it a different way. The branch between Drosophila grimshawi and Drosophila ananasi, which actually, as you can guess, grows on ananas, like pineapple. So grimshawi and ananasi are basically correlated when you look at the absolute lengths that you measure. But if you look at the normalized lengths, they're almost uncorrelated. And you can see that on average, the correlation is 0.6 compared to 0.01, 0.1 for the normalized string length. In other words, there's this common force that's driving all of these correlations. And you can think of that force as simply the fact that a gene evolves at a given rate and a species evolves at a given rate. And when you look at them together, they're just the product of the 2. Everybody with me here? Awesome. So what can we do? We can basically model the evolution of individual genes as sampled from some inverse gamma distribution, and the evolution of individual species, their relative acceleration, if you wish, as sampled from some gamma distribution. And therefore, the branch length is simply these, properties together. There's a gene rate, there's a species-specific rate, and then there's the time elapsed. Everybody from here? Can I get a 5, 4, 3, 2, 1? Awesome. Now, suddenly, that allows me to completely change the phylogenomic pipeline. So before, I would just do an all-by-all blast. I would cluster, I would align, I would build the trees, I would reconcile, and then, I would have some kind of reconciled tree. But now, if I have the species tree, I can actually do much better at tree building than that. Why? Because I now have a probabilistic model that basically says I'm given a species tree. I'm sampling topologies from within that species tree. When I have the topologies, I'm sampling rates. And when I have the rates, I'm sampling sequences. Okay, so it's a generative model that explains every aspect of this duplication, loss, evolution, topology, and mutation process. Everybody with me here? Cool. And that suddenly had this, extraordinary improvement in accuracy. So basically, if you look at neighbor joining, it had 60% recovery of orthologs. We now had suddenly 96% recovery. Neighbor joining would infer 22,000 duplication events. We could suddenly infer, 5,000 duplication events, and so on and so forth. Okay? Is everybody with me here? So suddenly we can get much, much more accurate trees that infer many, many fewer duplication loss events. Okay? All right. So all of that is modeling gene duplication and loss. But there's another component, which is that evolution doesn't happen in the whole species separating at the same time, but there's a diversity within a species. Remember before when I said that the mouse is carrying a bag of 20,000 genes and the rat is carrying a bag of 20,000 genes, and as soon as they speciate, all of their genes speciate at the same time? Well, in practice, it's not exactly that. And the reason is that species are made out of populations of individuals. And there's variation between that population. Specifically, there might be a duplication event that happens in the common ancestor of all of the mammals, and the species that carry that duplication event have some variation. Some individuals have it, some individuals don't. And that standing variation might persist along a speciation event and another speciation event. And now the standing variation is finally resolved when you get to a lower lineage. So all of that is thinking about evolution going forward. But when you run evolution forward, you have an exponential number of possibilities. Instead, you could say, well, let's run evolution backwards. I know that I have observed these 4 sequences. The question when you run evolution backwards is, when do they coalesce? And coalescence is when they meet up on the path to the common ancestor. Okay? And here you have regular coalescence, but then here you have deep coalescence. What does that mean? That means that the topology of this tree is incongruent from the topology of the species that it came from. In other words, if I look at A and B, they coalesce after C and D coalesce. Do you see that? So on the gene tree, C and D meet up first, and then they meet up with A and B. But in the species tree, A and B meet up with C first, and only then they meet up with D. Do you see that? This is an example that, if you look at backwards in evolution, is known as deep coalescence. In other words, these 2 lineages, the AB lineage and the C lineage, don't coalesce until after the D lineage. So here you have regular coalescence, and here you have deep coalescence. Everybody with me here? Conversely, you can think of this going forward in evolution as incomplete lineage sorting. That basically means that these lineages are sorting here, but they haven't completely finished sorting when the next speciation event happens. Is everybody with me here? Who thinks this is kind of cool? I hope you guys are enjoying this as much as I do. So this is like, understanding the subtleties of evolutionary processes. So now we have a problem because the same tree can be interpreted in two different ways if you only care about coalescence and if you only care about duplication and loss. If you only care about coalescence and you see an incongruent topology, oh, no problemo, that's a deep coalescence event. But if you only care about duplication and loss and you see deep coalescence, you say, oh, well, that's a duplication and loss event. Is everybody with me here? So now the question is, how do you model both together? And again, with my student, Matt Rasmussen, we basically developed this two-step reconciliation event that basically says I have a coalescent tree that evolves inside a locus tree, which in turn evolves inside a species tree. And I sample loci, where here I have a locus, which was duplicated. And then I can have my coalescent tree have deep coalescence within that locus. So suddenly I'm able to model both deep coalescence and duplication and loss in a single joint model. Everybody with me here? Awesome. Yeah. Beautiful question. And that's the topic of part 3, where things can go, all over the place. Okay. So the last thing that I want to deal with is this recombinations and this concept that recombination events effectively mess up the tree with which regions have evolved. In other words, I have inherited now a gene from my mom, who was here, and a gene from my dad. But my sibling has inherited a gene from the paternal grandma and the maternal grandpa, and so on and so forth. Is everybody with me here? So in other words, the moment I include coalescence events, up a tree in a population, and the moment I include recombination events, then suddenly I have the possibility of switching from position to position in the genome the topology of ancestral recombinations that, or ancestral coalescent events that explain any position. And that's known as an ancestral recombination graph, or an ARG. That basically tells you that these genes in the human population at this locus have this topology. So basically, very often we say, okay, what's the ancestry of this person? But then I'll ask you, yeah, but at what position? Because the ancestry of, say, one person in the class at this gene here might be, I don't know, Asia, Africa, and you name it. In that other position, it might be a different ancestry because they inherited a different gene from their parents. Does that make sense? So the moment I have recombination events, I can't just talk about a single phylogeny relating all people in this classroom. I have to think about all of the possible topologies based on all of the recombination events that have separated us. Everybody with me here? So then you can build this ancestral recombination graph that goes through all of this. All right. So the last thing that I wanted to leave you with is sort of this evolution near and far question. And sort of I'm not gonna talk about how we build alignments or how we infer these, but I will tell you about this very strange phenomenon where if you compare the species, the genomes of 4 closely related yeast species, you end up with more or less a straight line. But with that 4th species here, you end up with a broken up line. And the key insight here is what could have led to that, where you can zoom in here and open it up. And what you can see is that at every location of this one species, you have a whole alignment spanning 2 complete regions in chromosome 7 and chromosome 10 of that other species. And the same thing happens at every one of these places, where basically you have some kind of collinearity here, but then that segment is also here, and that segment is also there, and that segment is also here, and that segment is also there, and so on and so forth. So at every position, you end up with a region of one species mapping to two different regions of another species. But then the way that this mapping happens is not that all of the genes are in both regions. There's only two genes that are in two copies, but all the other genes are just simply interdigitated with each other. So what goes on here? What goes on is that there was, in fact, a complete duplication of that segment, after which this gene, that gene, that gene, that gene, that gene, and that gene were lost in one copy. And then this gene, 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 and that gene were lost in the other copy. Is everybody with me here? So when you see this interdigitation, it basically means that there was, in fact, a complete duplication followed by loss. Is that cool? So this actually happened to me. I basically was staring at these sequences, and I was trying to figure out what the heck happened. And then what we uncovered is that this is an example of complete duplication followed by loss. And that's not true in just one region, but it was true in every single region of the Saccharomyces cerevisiae genome, of Baker's yeast, the model organism that everybody has been studying for, decades and decades, is in fact a descendant of a whole genome duplication from a species that we just happened to sequence, frankly, because they sent us the wrong sample. We had asked for 4 closely related species. They mixed up the bottles. They sent us a further species. And thanks to that mistake, we're able to uncover a whole genome duplication event. And what's really extraordinary is that in some locations, you don't have a single gene that's maintained in 2 copies, because the vast majority of genes were lost after that whole genome duplication event. But in this region, you also see that the centromeres were in fact preserved, and that there's a 1-to-2 mapping between every pair of chromosomes, between the ancestral pre-duplication gene order and then the derived post-duplication gene order. Of course, both species are alive and with us today, but Cruvermyces walti represents that ancestral gene order. And you can see here that there was a gene here that was lost, and there was another gene here that was lost. And we can now study dramatic episodes of evolution using that. Okay? So that allows us to now reconstruct the ancestral gene order, but also infer that there was a whole gene duplication followed by gene loss, even though only 8% of genes are actually maintained in 2 copies. We can still unambiguously argue that there was a whole genome duplication. And it also allows us to now infer the fate of genes post-whole genome duplication. So we had 5,000 genes before, we went to 10,000 genes, and the vast majority of them, 90% of them, were actually lost in the second copy, retaining only 500 duplicated genes. And what's really cool now is that we can ask, given that routing from that common ancestor, which copy evolved faster or slower? And in some cases, we're basically finding that there was this dramatic acceleration event giving rise to new functions, that the silencing protein SIR3 evolved from a duplication of the origin of replication function that was there before, or this antiviral defense evolved from this translation initiation function, and so on and so forth. And what we found is that these derived function genes were never lethal. They were very specific in their expression, and they were specific in their localization. Okay? So that's, I think, where I'll have to stop today. But there's just so much. I'm going to leave you with one final story of the RNA world, okay? We all study all these species, and, we love everything about them, and we love the central dogma of DNA makes RNA makes protein. But when you look a little more deeply, you basically see that RNA actually has a lot of functions. RNA can basically play roles of, translation. It plays roles of splicing. It plays roles of catalysis. It plays roles of sensing and, ribo switches and all kinds of stuff. And that points to this ancestral world where RNA invented proteins, and together RNA and proteins invented DNA, okay? So that there was once an RNA world where RNA was used for information storage through self-replication. It was used for modifications, and it was also used for catalysis, where everything in that world was carried out by RNA. And eventually, there was this specialization of functions where DNA took over the boring task of storing information. Proteins took over the extraordinary versatile task of folding in 3 dimensions and carrying out functions inside the cell. And RNA, at least, still serves as the intermediate between DNA and proteins, but it also plays all kinds of other functions, okay? And that RNA world basically is one where RNA basically invented splicing, and you can still see that a lot of splicing is made by RNAs. RNA invented proteins, and that's the most extraordinary machine known as the ribosome, where you have this giant machinery where you have at the catalytic core lies only RNA. It's a combination of RNA and protein, and people thought for a long time that maybe, the catalysis happens by the proteins. But the structure of the ribosome basically showed us that the catalysis is all based on RNA, that proteins were only added later in evolution to stabilize that whole structure. And then the adapter molecules themselves are tRNAs, which on one side function as DNA by basically binding by complementarity and reading the triplet code. And on the other side, they function by structure, bringing together a particular amino acid for a given transfer RNA molecule. So that extraordinary invention basically allowed now RNA to invent proteins. And what you see is that in all of the operations that involve DNA, RNA didn't bother inventing DNA by itself. RNA first invented proteins, and then together, they invented DNA through reverse transcriptase. So basically, transcription itself happened only later, but reverse transcription came before transcription. And reverse transcription was basically the process of taking this RNA information and storing it in a more stable molecule. And then together, they make all kinds of additional functions, okay? All right, with this, I will leave you with this extraordinary topic of evolution. We only have one lecture, unfortunately, given how much other stuff we want to cover, but hopefully gives you some glimpses of this extraordinary diversity of first understanding phylogenetics, the basics of phylogeny, going from alignments to distances, from distances to trees, and from alignments to trees, and this tree exploration. The complexity that arises from understanding that genes evolve inside species and this phylogenomics approach, and also evolution in populations in complete linear sorting, and these extraordinary events, these evolutionary jumps that can happen by whole genome duplication. And then the slides that I kind of skipped in the middle are about the evolution of evolutionary thinking itself. Basically, this introduction of natural selection by Darwin, and the advent of genetics with Mendel, and all of these different discoveries. And I really encourage you to just spend a little bit of time studying that slide, because it's still an evolving field. As you could see, like even students like you can sort of have an impact in understanding how to bridge together all of these different types of events. But it's truly an extraordinary field, and I hope you guys go on and study it. All right, that's where we'll stop here, and we'll continue on Tuesday after Thanksgiving break with metabolic modeling. So thank you all. Have a nice Thanksgiving break.